I came across in the literature in recent times was these glucose spikes markedly enhance the gut mucosal permeability.
Yeah.
And so it sets up for inflammation, and so also vulnerability to infection, and is even work to suggest a predisposition to cancer. Had you had the sense that the microbiome was going to be an important driver?
When we started the study, so we had this hypothesis that, okay, we decided to focus on blood glucose levels. We had this hypothesis that people are going to have different responses. So kind of thinking one step ahead, we said, well, let's assume that that will be true. Then we'll want to, our next question will not be just to, okay, demonstrate that there's individuality, but we want to hopefully develop an algorithm that would be able to predict that. So let's collect everything that we can about a person. And in the research, we went very broad. And then one of the components was the microbiome, because, so the whole field of microbiome is very young. It's maybe a bit over 10 years that it really picked up with the advances in sequencing and finally the ability to sequence our bacteria. And so this is maybe 7 or 8 years ago, so the field was maybe 3 years old, but still there were some seminal works coming from Jeff Gordon and Rob Knight, who's going to speak later here today, really showing the, not just associations, but the causal effect of the microbiome on metabolism and obesity. You know, these seminal experiments where they take microbiome of obese, even from identical twins, taking their microbiome who are discordant for obesity, and showing that if you take from identical twins the microbiome of an obese person, you put it in a mouse, the mouse in 2 weeks gains much more weight than transplant taking the microbiome from the lean person. So all of that indicated to us that perhaps on the microbiome is going to be a very critical marker, also in terms of its causal effects.
There's kind of shortcut way to assess it, which is used by a lot of commercial companies now, and then there's the true metagenomics. And can you comment about why one is different than the other? Because I think a lot of people are not up on that.
Okay. Yeah. So so that's also a very important point about how we look at the microbiome. So in the early days, the way people looked at the microbiome was by looking at a single gene. It's the 16S ribosomal RNA gene, and the reason, and there was a lot of good reasons to looking at that. I mean, sequencing was still a bit expensive, and it's one gene that it's a gene that has some very conserved regions, virtually identical in almost all bacteria, but that same gene also has some variable regions that vary across certain bacteria. Some different species will have variability. So what that allows you to do is to use PCR. So PCR is a way to amplify DNA and use universal primers that will amplify that gene across all bacteria, because you amplify from the conserved DNAs. And when you amplify that, you can sequence the variable region, and then you can look at the composition of bacteria that you have. So it was a very good idea, and this is how people have been looking at it, and it's much cheaper also to do. And that kind of gives you, but if you think about what it gives you the overall composition. It has many limitations because although there's that variable regions, there are some higher order, in some cases even an entire genus, which can have thousands of different species and of course millions of different strains, will all be identical in that variable region, so that method will not be able to differentiate that. Of course, the whole microbiome is it's very complex. If you think about it, it's like our genome when we all have the same Homo sapiens genome, but it's only true up to 99% identity, and the 0.1% across the 3 billion base pairs is several millions of differences between each of us, and that really is all that's the genetics that we study. So bacteria are the same. When we all have, many of us have E. coli in our gut, but that, if we look at the genome of E. coli, which is about 4 million base pairs, we're going to have a lot of variation between people in that E. coli. And some of that variation can have tremendous impact on the function of the bug. So only metagenomic, when you do metagenomics, basically what you're doing is you're just extracting all the nucleic acids, you extract all the DNA in the sample, and you sequence everything that's there. That's called shotgun metagenomic sequencing. So then, only with that method, you really have the resolution and the ability to look across the entire genome. And I presented a little bit of that today. We have a paper coming up showing that if you look at the same bacterial species across people, you will identify thousands of different regions in which there's some regions that are deleted in only some individuals and not in others. And that itself is in association with certain metabolic disease risk factors. So that's really an entirely different resolution that you can study.